The overall goal of this experimental procedure is to examine the impact of specific host processes on the ability of bacterial pathogens to translocate effector proteins.
This method can help answer key questions in the field of cellular microbiology, specifically how the host is involved in either blocking or promoting the delivery of bacterial virulence factors into the host cell. The main advantage of this technique is that it combines two established techniques, namely siRNA gene silencing and a reporter assay to measure bacterial protein translocation, to investigate the interplay between pathogens and the eukaryotic cell.
This method provides insight into Coxiella pathogenesis and can also be applied to investigate the host pathogen interactions of other bacteria that depend on efficient protein translocation. For example, chlamydia, salmonella, and attaching and effacing E. coli.
After culturing C. burnetii expressing CBU0077 fused to beta-lactamase according to the text protocol, in a 25 cm² culture flask with a vented cap, inoculate 10 milliliters of pre-warmed ACCM2 containing 3 micrograms per milliliter of chloramphenicol with 20 microliters of the bacteria. Grow the culture at 37 degrees Celsius with 5% carbon dioxide and 2.5% oxygen for 7 days. To reverse transfect HeLa cells with siRNA, and after preparing the siRNA according to the text protocol, place DMEM plus 10% FCS, 0.05% trypsin EDTA, and PBS in a 37 degrees Celsius water bath for 30 minutes to warm prior to use. For each experiment.